welcome to your second lesson of the week. We'll start straight away with our 6 questions. The first one of which is define selective breeding for yourself. Okay, we covered that last lesson, so go back and watch that video if you're not sure. 2, what is meant by the gene pool? 3, how does variation provide evidence of evolution? 4, name me 2 types of pathogen. 5, what type of bonding is shown in this diagram here? And 6, describe how you could calculate the speed of an object. Okay, good, let's go for our answers. So selective breeding is about breeding for desired characteristics, the characteristics we like best. Gene pool is all the genetics, all the genes available for an organism, so all the potentials for variation. And that variation provides evidence for evolution as it shows that an animal, an organism, a species is adapted to its environment and its surroundings. The 4 types of pathogen you had to choose from are protists, fungi, bacteria and viruses. That type of bonding shown in the diagram is metallic. And then calculating the speed of an object, you'd get a mark for stating that speed is equal to distance divided by time, and then your other marks would be for describing how you'd get that distance in that time. So you'd measure the distance using a tape measure, and then you'd record the time taken to travel that distance using a stopwatch. Okay, so last lesson we looked at selective breeding and how we are able to use that to promote desired characteristics or to try and remove undesirable characteristics. And today we're going to look at how we're able to predict inheritance and to predict whether we're likely to pass on our desired or undesirable characteristics. And to do that, it involves drawing out something called genetic diagrams. And so I'm going to do a fair bit of that today. But beforehand, we just got to remind ourselves of a couple of facts as well as introducing a couple of new words. So the first thing to remember is that we have the potential to have 2 versions of each of the genes in our body because we get a copy of our mum's genes and our dad's genes. And those 2 genes could be exactly the same. So we could receive the gene for brown eyes from our mum and from our dad. And when those 2 genes are the same, we say that we are homozygous. Homo means the same, so homosexual means attracted to the same sex. But equally, we could get a different gene from our mum and from our dad. And then we have 2 genes or alleles that are different for hair colour or eye colour. And when this is the case, we say that we are heterozygous for that allele. Hetero means opposite, so heterosexual, you're attracted to the opposite sex. Heterozygous, you have 2 different genes. Even if you have 2 different genes like this, you can only ever see one of them. One will win out, and the displayed gene, the displayed characteristic, must come from the dominant gene, whereas that other gene becomes recessive. It sits there in the background and isn't seen. So my hair, for example, I have a dominant gene for ginger hair, which is why I'm ginger, but I probably have a recessive gene for a different hair colour floating around somewhere in the background because neither of my parents have ginger hair. And these recessive genes are able to be displayed and become dominant, or at least shown, depending on what your partner has in their genetic makeup as well. And so this is why genetic diagrams are useful. They allow us to look at how these genes come to the forefront or indeed can be avoided. So we're going to have a look at a little example of this, and we're going to look at an example of how we get the perfect carrot, because I've drawn these lovely pictures of a carrot and I may as well use them. So this shows how genes can be passed on in a plant before we go on to look at some other examples. Okay. So the point of genetic diagrams is they allow us to see the likelihood of a particular trait being inherited or not. So if we think of 2 carrots here, and one of them is a nice straight perfect carrot and the other one is a bit wibbly wobbly and a crooked carrot, we are then able to think about these 2 carrots and the genetics which cause this difference in appearance. Okay, so one must have a gene or an allele to make it straight, and the other one must not have that gene. So they've both got two alleles for their shape. The straight one has a different one, and we show these by these letters here, a capital S and a little s for the straight caret and two lowercase s's for the crooked caret. So the crooked caret has two alleles the same, so we say it's homozygous, and the straight caret has different alleles, so it's heterozygous. And we use a capital letter to show a dominant allele. So the straight caret is straight because it has that dominant capital S, which means just by having that, the caret will always come out straight. So then a genetic diagram is just a way of showing all the possible combinations of these four alleles in total, two from each parent. So you split them up using a grid like this, the capital S and the little s from one caret, and then two lowercase s's from the other one. And then you combine them. So you can have big S, little s, two little s's, another big S, little s, and then finally in the bottom right, two little s's. And this is just allows us to then predict the likelihood of the offspring, in this case the carets that are produced, the chances that they'll be straight or crooked. And we use this same idea to be able to predict crossbreeds and things like that in plants. And because it's 50-50 of each, there are two with big S's, there's 50% of them straight, 50% crooked. Okay, so as important as it is to have the most beautiful vegetables possible, it's generally more important and more useful to use these genetic diagrams to see the chances of inheriting genetic disorders which may cause ill health. So we're going to have a little look at an example now which goes through that. So an example of one of these genetic disorders that you might want to avoid inheriting is something called polydactyl disorder, which you can just about see me writing at the top here. And this is a disorder whereby which you might have an extra digit on your fingers or toes, and it's caused by a faulty dominant gene. So if you get a gene, one and two alleles for this, you'll definitely display this disorder, this extra digit. And so you'll often get a question asking you about the inheritance of a disorder like this. So it says here, two parents are heterozygous for polydactyl disorder, or for the disorder. And it really doesn't matter what the question is, what it's about here, what disorder it's about. The key bit is where it says heterozygous. That's the bit that gives you a bit of information about exactly what's going on. Okay, and then it'll ask, how likely is it for the offspring to inherit this disorder? And as I said, it's heterozygous. That's your key word. So you need to be saying or remembering that heterozygous tells us that the two genes, the two alleles are different, that you'll have one for the disorder and one regular gene not for it. And remember, we show these two genes by different letters, and the capital letter is the dominant one. And for the two parents to have this disorder, to display it, it must be that polydactyl is caused by a dominant faulty gene. That if you have it, it will always display, it will be sitting on top. Whereas that other gene, the regular gene for 5 digits, is a regular recessive gene. So then we can use one of our genetic diagrams to predict the inheritance for this. So just as we've done before, it's just a case of drawing these grids and then splitting up my two alleles, my capital P for my dominant polydactyl gene and my little p for a regular gene. And then we can combine these. So my two big P's together, my big P and my little p, another little p and big P, and it doesn't matter which way around you draw them, and finally two little p's. And it says then, how likely is it for offspring to inherit? Well, these three with the big P, because it's dominant, that child, that offspring, would have the disorder. So 75%, 3 in 4
would display polydactyl disease, they would have it. Whereas that final one, that 1 in 4, 25% chance, wouldn't. They wouldn't inherit the disorder. And the final thing to look at here is this one, as I've circled here. This is homozygous for it. And that offspring, that child, would definitely then pass it on to their offspring.
You can use these genetic diagrams to show why it is that we've got a 50-50 chance of having a boy or a girl when we have children. And it's because the gender of a child is determined by chromosomes. And female gender means you have XX chromosomes, male gender means you've got XY chromosomes. And every sperm cell has that XY combination, every egg cell has that XX combination. And so when you combine an egg and a sperm together, you get a mix of XX and XY chromosomes. And when you fill in one of those genetic diagrams, which we'll do in just a minute, you see that you end up with a 50-50 chance of a boy or a girl. And that's us just about done for this lesson and this week. No exam question for this topic because you very rarely get big questions on it. It's more about being able to fill out those genetic diagrams. If you do want to have a look at another exam question, there's one on the extension task. If not, have a good week. Thanks very much for watching. Bye-bye.